Hi, welcome to National Science Day Lecture Series 2024 on applications of biotechnology for Vikshit Bharat webinar and quiz competition. This is Dr. Karthikeyan Ramalingam, Professor, School of Life Sciences, B.S. Abdurrahman Crescent Institute of Science and Technology. So today we're going to talk about important topic, nanoemulsion, a game changer for sustainable agriculture. First of all, we we'll know about introduction of nanoemulsion. Here, when you see nanoemulsion, that is, for example, is it possible to mix water and oil together? So we imagine that oil and water, when you mix together, it won't mix properly, right? So here, this process, what we are doing it, we are mixing oil and water with the help of surfactant. When we add surfactant, so the oil and water mix and make a tiny droplet. So that will be very stable. You see here, the end here, you see this oil look like a droplets. So here is a nano emulsion. We are making a nano droplet using a method. So nano emulsions are thermodynamically stable isotropic system which has two immiscible liquids are mixed to form a single phase with the help of surfactant, that is emulsifying agent. So what is the advantages when you talk about nano emulsion? See here, this first the beaker, you can see the true emulsion that's uniformly formed. But when we mixed, if without a nano emulsification process, you always see the the creaming will form, or flocculation will happen, or sometimes sedimentation will happen, or separation will happen. So these kind of things we can avoid when we make a nano emulsion. So what is the advantages when I talk about, as I told before, the nano emulsions are thermodynamically stable, and also based on the composition it's non-toxic and non-irritant, even it is taste masking in nature. So nano emulsion can be administered by the mean of oral or topical or transdermal. Different delivery is possible with by when we using nano emulsion. So both we can also deliver hydrophilic compounds and hydrophobic compound also, because both oil and water option is there, where based on the nature of the compound we can use the solvent, any of these two compounds for mixing the compounds or drugs. So nano droplets, obviously it is increase surface area, and this will enhance bioavailability, and also is protect drug from hydrolysis, oxidation by encapsulation inside the droplet, and also it is helps enhance permeation of drug through the skin also because of its nature. So nano emulsion can be prepared different methods. For example, nano emulsion can be produced by high energy method or low energy method. So if any talk about high energy method, we have high pressure homogenizer, or we can use ultrasonication method or microfluidization method. So commonly we use a microfluidization for preparation of nanoemulsion. If it is when you talk about low energy method, we can use a phase inversion or solvent displacement or spontaneous emulsification methods. So we generally use a microfluidization method for preparation of our nanoemulsion. So after preparing the nanoemulsion, we need we can use a different way of characterization of nanoemulsion. For example, generally what we do characterization to check the property of nanoemulsion, we can check the dispersion, you know, types, how it is disturbing, dispersing, and also we can easily check pH, and we can also observe light microscopy. You can also we can use a Sudan dye and different dyes we can use to stain the droplet to see clearly in the light microscope. And also viscosity can be measured by using viscometer. And very importantly is very commonly gold standard method. We can use a DLS, dynamic light scattering, and zeta potential. Only these dynamic DLS and zeta potential, it will show you, the property of nano droplet. Is it stable or good or bad, and what is the size and everything we can get it from the DLS and zeta potential. So after characterization, when you talk about generally application wise, so different applications nano emulsion in used, for example, cosmetics or agrochemical or food industry or healthcare or personal care. Different applications are there for nano emulsion. So when talk about oil and water nano emulsion, what are the how you supposed to do it? For example, here we need a water and we need a oil, and also we need a surfactant. And usually we in our formulation we use a cation as a compound, active component. So these all four together we need to mix, and this will go to the microfluidization process. For example, this is LM10, a microfluidizer we used to use for our experiment. This is company, US based company, this instruments of manufacturer. So this can produce 20,000 psi pressure. With that pressure, the all component will be you know have a interaction. All the component happens in the interaction chamber of this LM10, and finally we will get a tiny droplet, nano droplet. So that droplet have a thermodynamically stable property. So it depends on the components or formulation, the working nature of nano emulsion will differ. So what generally the oil source, the nano emulsion composition, for example, oil source, multiple oil, for example, whatever different oil, if it is can be essential oil or it can be a castor oil or it can be a soybean oil, a different oil we can use it for this preparation. And surfactant, examples of surfactant, for example, Bridge 30, Triton X, Twins 60, or Span 20, and different surfactants are there. We can use it to preparation of nano emulsion. It's all depends on what for you are making the nano emulsion. Even many people usually use a co-surfactant also. For example, PLG, propylene, polypropylene glycol, or twin span 80, glycerol. Many people use you know, isopropyl alcohol also as a co-surfactant. So composition wise, this actually we referred more than 100 papers. We come to a conclusions. What are the different the surfactant, co-surfactant people have been using it. Okay, so now we have prepared the nano emulsion. When you see the appearance of the nano emulsion, this looks like this, the milky white. Okay, these are some of the formulation what we used in our lab. Okay, so now you know the appearance. You may have a question in your mind. What is the mechanism it is involved with the nano emulsion? What we propose the mechanism, because most of the nano emulsion we use for antimicrobe as an antimicrobial agent. So based on our results, so we come to some conclusion. Nano emulsions, as I already told you, this droplet produce very high pressure. So the droplet, for example, this is a nanoemulsion appearance. As I told, this is a microorganism. Microorganism, this negatively charged cell wall is there. So nanoemulsion here, this droplet is positively charged, because I already told you we add some positive charged ions also for the preparation of nanoemulsion. So the charge-based attraction will happen with the nanoemulsion. So after the charge-based attraction mechanism, this nanoemulsion attached to the the cell wall of bacteria. So after attaching it, it is enormous pressure inbuilt in this nano droplet. It may have explosion effect. See, you can see the like bombing effect, because as I told, the pressure built inside the droplet, it can burst and it may damage the cell wall of the bacteria. So since this mechanism, because of this mechanism, we always say this is a non-resistant mode of mechanism, because nowhere it is any compound is involved, only pressure built inside the droplet. It is making the issue for the bacteria. So how, maybe you may have some question how you are confirming it. See, we have done multiple studies. For example, this is one study done with Acinetobacter baumannii. It is one of the pathogenic organism. It caused a lot of problem in the Iraq war also. See here, this one strain one. Okay, when we see, this is a control how beautifully they are made a biofilm. See this, the pili formed. Even here also you can see very nicely they have performing fili and then communicating each other. Very nice biofilm, healthy biofilm. But when you see here the treated with the nano emulsion, in very bad shape. You see that completely, irregular shape and damaged look you see. And then even some other microbes. This is actually, other different strains. You can see that these are all control, different nanoemulsion, different strains, and then nanoemulsion treated. You can see very clearly. Even you can see that, you know, the cavity is formed in the treated one. So, so based on this, even we have done some more assay. For example, this actually, we used a live dead strain. Live dead means the live bacteria will be shown in the green color. A dead bacteria you can see in a red color. And this is actually for this experiment. This is all control. You can see very greenish in nature. Okay, and then nanoemulsion one dilution, you can see very intense activity. You can see in this panel of confocal images. But when you dilute this 500 dilution of this nanoemulsion, It is reduced the activity of in the live dead cell number is reduced in this panel. You can see very clearly. So even that, you can see even this is some more other microbes also we tried. This is actually a streptococcus mutans biofilm. You see very beautiful control cells. But when you talk about, when you see in this treated, see completely disintegrated, no margin at all. These are all the, the droplet, the bigger droplet of nano emission. Okay, so these are all droplet also captured in the ACM images. So one time, okay, somebody asked the question, how do you say this is nano emulsion is doing the work? Why not this component? So we tried, always when we do our experiment, we have a control, okay, without nano emulsification process. And also we used to do separately the ingredients. You can see this one formulation we use Triton X100. It's a surfactant, CPC as one of the core surfactant, combination of these two, and then finally nano emulsion. You see the big difference. You can see this nano emulsion, reddish color. That reddish color means it is, lot of cells are dead. When you compare these other ingredients, you can clearly see that, the nano emulsions working better. And this is some of the nano emulsion. We multiple formulation. This actually, this is we done for two of our patent. This actually I'm going to discuss about two of our patent nano emulsion formulation here. You can see this different nano emulsion and showing the the DLS, the dynamic light scattering of these nano emulsion. So even here also different nano emission. You can clearly see that size range. It's around 100 plus 200. So I can show you some more table of difference. Also nano emission. For example, here you can see here. So size range of nano emission 133 and then 173 and 129 and see different. These are all the commonly, common sizes of nano emulsion when we prepare. Even we try to use this nano emulsion to check this toxicity in a different way. One way we use Bombix mori, the silkworm, to check this toxicity of this nano emulsion. So you could see, more healthy uh, Bombix moris are here. Now, after some time, after 12 hours or maybe 20 hours, you may see some the unhealthy uh, Bombix mori, like, silk forms. Okay, so up to now we discussed about what is nanoemulsion, what is advantages of nanoemulsion, and also how what is the methods are being used for nanoemulsion synthesis and characterization, and also mechanism proposed by us, how it is killing the microbes since it is a antimicrobial nanoemulsion. So now, as an important topic, like what way it is helping in, you know agrochemical, how it is helping for agrochemical. Even many studies are available. Even people used thymol nanoemulsion, okay, for controlling. It's used as agrochemical because thymol, it is already known antimicrobial compound that can be antibacterial, antifungal, and even it is help for plant growth regulations. So this can be these already people have used as a biocontrol agent. So now I'm going to discuss about what we have done in our lab, for to check this nano emulsion. Is it how far it is working in agricultural problems. This is one of the study we used aspergillus flavors with like peanuts. So peanut we have used aspergillus, infected the aspergillus, both seeds and also leaf, and then we treated with the nano emulsion, and then it was the appearance, and also the you can see here, this is a control A, this is a control completely. The aspergillus flavus is grown very rapidly. In others appearance you can see is completely no sign of aspergillus flavus. And same was confirmed by microscopy and also other assays, like you can see clearly the nano emulsion is not allowing the the aspergillus flavors to grow. And here even we checked after this infection study, like like we have even tried to grow these peanuts. It was growing good, like a control. So seed germination happens, normally, even after using nano emulsion treatment. So what I'm trying to say here, so nano emulsion is not you know hindering the seed germination. Similar way, even we tried with the pyracloria oryzae in the rice and paddy. So similar kind, the results we have got. And then, so this is for rice and other plants. Later, we want to check, is this a nano emulsions? Is it extending the life of perishable fruits, for example, tomato, strawberry, and apple? So we have made different nano emulsions, and then different dilution, for example, 1 is to 1 or 1 is to 100 dilutions. And then that we have used with the tomato, for example. You can see clearly here, mostly we have done different in vitro assays. I'm directly coming to the real time study what we have done under room temperature and also refrigeration. You can see very clearly, see day 1, day 2, and different groups, different nano emulsion spray. For example, next, for example, this is day 3 and 4, and then day 5. You see is some changes maybe you can see in the control, but no changes in the other groups. You can see, for example, other day 8 and 9. For example, now you are seeing 12. After even 2 weeks, you can see here is 2 weeks of the A, it is started shrinking. The skin is shrinking, but you see others, they are all still looking fresh flesh. For example, again, this is 17th day, under refrigeration. After 3 weeks, see, still you see the tomatoes are looks fresh. And even we tried, this is I'm saying this this is a way the nano emulsion extending their life of tomato. So when you talk about the control, see this control here, and even here after 3 weeks, you can start to shrink, and then a lot of problems come. I'm not telling all the formulations working excellent. Some of them very good. That's what I'm treating. Very comparable to a normal exposure without nano emulsion. This will keeping the tomato very healthy and fresh. That is what I'm trying to say. For example, even we tried with the room temperature. You can see room temperature, definitely you see a big difference. See day 1, day 4, and then here you see after a week time. See, see here completely, the after 2 weeks you see this room temperature one is completely gone. Even one of the nano machine is not working good, but others, see, It looks very fresh, same as the first day. So we surprising these nano machines make doing wonders. So that's the graph is showing how effective even both refrigeration and also room temperature. So nano emulsions are working excellent. Same way, Same way we tried with the strawberry also. See, I hope you may everybody know, when we buy strawberry, even if we put it in the fridge, it gets spoiled easily. But when we spray this nano emulsion, it is showing 3 to 4 days. And if it is refrigerated up to 1 week, it is showing a fresh and healthy strawberries. We can try that. Maybe you may ask, how about is it good, or the nanoemulsion is good or bad, because we are spraying in the fruits. I always say the formulation what we make, that even you can drink, that much safer nano emulsion what we making it for, if I use with the food and eatable or animals. So even we tried with the apple, the same kind of results we have seen. Even some this actually we displayed in some exhibition. Somebody told, can we do it in the cut vegetable? We tried in the cut vegetable. It's nanomesh help to extend the cut shelf life of cut vegetable also. And so what I'm trying to outcome of this project, this nano emulsion demonstrate as very good life extension of tomato or strawberry, even apple, and even cut vegetable. So even this nano emulsion, it will be a cost effective one, and also it is in a good edible nature. So 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 our lab, we do uh, different types of nanoemission based work. For example, we try antibacterial, antifungal, antiviral. Even we recently we have made a pesticide detox nanoemission. We multiple students worked in this project, and then we use this nanoemission for preservation of meat, vegetable, fruits. That's what we discussed some of aspect. Even we tried a different formulation for bacterial growth enhancement and shrimp growth enhancement nanoemulsions, different nutrients we deliver through nanoemulsion, and also some formulation we use it for pathogen management, some vibriosis management in the shrimp, and even ornamental fish with pathogen management. Major work we have done with escape pathogen. Uh, this picture you can see, this is Dr. Asim Khan. He done his PhD work in this escape pathogen by using nanoemulsion. Mouth rings we have developed. Still we are working on for final commercialization process, the cryogenic nanoemulsion, and even now any we finished with the nano emulsion, we are how next delivery we are working on it. So we nano emulsion based ointment we are developing right currently, and this nano emulsion can be work as a biofilm inhibition agent. And even we have recent patent, we had a nano emulsion with a different nanoparticle. We have the using it and to enhance the activity of nano emulsion or nanoparticle also. So nano emulsion, we work for a targeted drug delivery or a cancer treatment, and pesticide use as a pesticide, or even wound healing process also. So multiple application our lab working on the nano emulsion. So this is a picture of our, group PhD research scholar. Is Doctor. He is now is Doctor Hasim Khan. She's Dr. Sneha, And these two people is Jahangir, and she's a Kirtana. These people are working in our lab right now, and some more research scholar also working in the similar kind of field. So finally, with this slide, I'm concluding this lecture. Like so, maybe I after seeing this, the complete view applications of agriculture. So definitely you may have some idea. Definitely, it's a game changer. The nano emission is a game changer for sustainable agriculture. So see this slide. You are the key to inspire you only. Okay, so other key doesn't work. So I would say work hard, you will get a outcome for that. So thank you for listening. Have a great science day 2024. Thank you.